hello everyone. I am Professor Dr. Ashlo Panje. I'm head of laboratory for colloid biology at the Department of Environmental Sciences at Jožef Stefan Institute, Slovenia. And today I'm going to talk about the development and construction of biocolloid structures, especially artificial biofilms and other similar constructs. So the first thing, we have to understand what are the boundary conditions for controlling cell-to-cell interactions. So if we want to establish such, let's say, structures, we need to use universal mechanisms that are working everywhere in the universe, should be applicable to all cells, then can be controllable, then must be possible to add other properties to these components, should not interfere, or at least should interfere at the lower extent with the physiology, and of course, if needed, can be triggered, that can be done in the interaction, should not be toxic, or well, should be adaptable or available mechanism to modify these structures, should be cheap and in high access, and at the end, should be measurable directly, indirectly, how this, let's say, controlling is made. So if we want to do all these things, these boundary conditions, we need to understand there are four important forces in the universe, and only one can be applicable to the biological organism, so electromagnetics. So if we take the electromagnetics, so the electromagnetics are governed toward the repulsion, the attraction, all this kind of physical interaction between different colloids. And because this is inanimate particles, the same, we can do it with the biocolloids, which are the alive bacterial cells. So how we can do that? So first we have to change their electrostatic properties. So changing from minus to plus, because we all know that the bacterial cells are slightly negatively charged. So we need to have or develop tools to do that, to change into the positive charge. So if we change this positive charge and we have the negative charge surfaces, the biocolloid particles get attached on that surface. And the same can be if we have the positively charged particle, biocolloid particle, bacterial cells, the other negatively charged will be attached to them. So the first thing, we have to modify the bacterial cells. How we do that? So we use the different polyelectrolytes that are charged polymers, and we put them on the surface of the cells. And this is the procedure, how we do it. It's relatively simple, but at the end can be very complicated. Depends on what sort of cell we have, because some of the polymers can be toxic, so we have to adapt that. And the conditions, physical conditions, how we can modify these surfaces can be different from cell to cell. At the end, we can produce certain capsule, which is in this picture red, and is entrapping the cell, which is in the green. So the good thing of these polyelectrolytes, it is that they are, in most cases, not toxic. They can put different charges. So as we can see, they are negatively and positively charged bacterial cells, and the surface of that is covered with very thin layers between 2 to 6 nanometers, and the distance between the polyelectrolytes are relatively large, forming large pores that can be exchanged different sorts of nutrients through that. So the concept is that what we see in nature, like this biofilm, that can be produced artificially, we should treat these cells as a Lego bricks, and to produce such kind of structure artificially, like biofilm, artificial biofilm. So, and we can tell this like a Lego bricks that we can put them together and forming such artificial structures. So what we did in the past was that we have three different pillars. One is bulk aggregation. We call it as a cake model because we can put these different cells in the aggregates randomly, or we can make very specific controlled way aggregation where we can put different cells in really advanced structures, or we can make structures something like biofilms where we put different sorts of cells in the layered position. Or at the end, if we put these cells in different aggregates or different structures and different cells all together, we can make very complex structures, something like big structure composed of different sorts of cells, where each cell is beneficial to the functioning of this aggregated structure. And of course, the physical properties of this structure, like amount of oxygen or amount of different nutrients inside these aggregates, is developing new niches. And new niches means the changing of the metabolic properties of cells. So something like this, as is shown here, the different structures or artificial aggregates can form the anaerobic zone inside or aerobic zone outside. So engaging cells in different metabolisms, as it is shown here with the Shewanella densis. And this strain is particularly capable of precipitating different toxic metals, in this case vanadate, as we can see in the lower pictures. And by precipitation, that has very high bioremediation potential. So another thing is that by such structures, we can stimulate the cross-feeding interaction between cells. So as we know, the distance between different cells is actually describing how they're interacting. So closer we put in a certain amount of aggregation, better we have interference between them, and cells can change from the competitive interaction to the collaborative interaction between different cells. Another thing is remote guiding of bacteria, where we can actually use the approach where we modify the surface to change the bacterial cells to the magnetic particles, and we can then put them into different position. So this, to produce that, and of course to modify the surface in the way that we can see it through the body of organisms, we can actually develop the mode of action where we can use the bacterial cells as in the anti-cancer treatment, directing them initially toward the cancer structures. Another approach is to make artificial biofilms, very complex artificial biofilms, where we combine inanimate polymers together with different sorts of bacteria, and can be really applied to the field of protection of the biocorrosion through the forming special sort of biofilm, preventing growing the biocorrosive strains over the surface of this metal. So, but not only that we want to make a stable structure, because someone can say, okay, how long can this structure last? Because these are alive particles, so they change, but also the complexity of the dynamics and stability of structures can be governed by these processes. So it means, as in this movie, that the small structures that are formed can engage cells to keep this structure and grow in the direction that we predict in the future. So thank you very much for listening me on this MOOC, and I hope you enjoyed these very fascinating results that we brought to you. Thank you very much.